So I did my poster presentation on BBS1 related Bardet-Biedl syndrome and its effects causing obesity. So there's 3 questions you have to ask before learning about my research paper. The first question is, what is the normal function of the BBS1 gene? The BBS1 protein is a part of a group of proteins that play a critical role in the formation of cilia. Cilia are microscopic finger-like projections that stick out of the surface of some cells. They are involved in the cell movement and many different signaling pathways. The second question is, what disorder is associated with the BBS1 gene? Bardet-Biedl syndrome is an autosomal recessive disorder associated with the BBS1 gene. More than 30 mutations with the BBS1 gene have been identified in people with this syndrome. Mutations in the BBS1 gene likely affect the normal function and formation of cilia. Defects disrupt chemical signaling during development and lead to abnormalities. Researchers believe defective cilia in Bardet-Biedl syndrome are responsible for the presence of vision loss, extra fingers and toes, kidney defects, intellectual disabilities, and obesity. My graph shows BBS1 is the largest contributor of Bardet-Biedl syndrome. It's most commonly found. It's actually 23%. There are other genes that contribute to Bardet-Biedl syndrome. BBS2 through 12, there are 2 other genes, and 21% is actually unknown. The last question to be asked is, what is leptin? Leptin is involved in regulating appetite and body weight. Abnormalities in the production of leptin are associated with alterations in body weight. My paper that I researched is the BBS-SOM controls energy homeostasis by mediating the transport of the leptin receptor of the plasma membrane. It was a series of experiments done. The first one was in this experiment, in order to first gain insight in the role of the BBS proteins in the nervous system in the regulation of energy homeostasis, a conditional knockout model where exon 3 of the BBS1 gene is flocked was used. Breeding of the BBS1 FLFL and nesting Cree mice were created, mice deficient in the BBS1 gene only in the nervous system. The loss of the BBS1 gene expression in the brain, but not the peripheral tissues. This is important that it was in the brain and not the peripheral tissues. At the time of weaning, which was 4 weeks, up to 8 weeks, the mice were weighed at the same time. At 9 weeks, the mice began to weigh more than the controls. At 25 weeks, the female mice weighed 53% more than the controls, and the males actually weighed 18% more than the controls. The fat mass were measured by MRI in 12 and 25-week-old mice. The weight was confirmed by fat pads, and there was no difference in lean mass by MRI. The food intake during the 5 days showed that obese mice ate 35% more than the controls. The next experiment they did, they used LRB Cree mice, which provided specificity of Cree expression to LRB expressing cells. They assessed whether LRB cells express the BBS genes. At 4 weeks of age, there was no difference in body weight between the LRB mice and the controls, but at 6 weeks, they started weighing more than the controls. At 25 weeks, the mice weighed 30% more than controls, and as they began to age, they became morbidly obese. The difference between the LRB mice and the original experiment was that the weight gain was similar between male and female. Fat mass and plasma leptin levels were not different in the young mice relative to the controls, but in older mice, they began to develop adiposity, as indicated by the increased fat mass, plasma leptin, and size of the adipocytes. The liver mass were indicated by the LRB mice were revealed in oil red O staining. They then did check the energy imbalance in the LRB mice. The food intake was no different between the LRB mice and the control at 4, 5, and 6 weeks, but at 6 weeks, the mice began to eat consistently more than the controls. They actually ate 20% more than the controls. The fecal caloric content showed that they had normal digestive efficiency. To examine whether hyperphagia can explain increased adiposity in the LRB, a pair feeding study was performed. At 25 weeks, body weight and fat pad masses of pair LRB mice were substantially lower relative to the LRB mice, indicating that hyperphagia contribute to the obesity phenotype of the LRB mice. Findings suggested reduced energy expenditure. The O2 consumption and heat production were lower in the obese LRB mice compared to the controls. They then tested for leptin resistance in the LRB mice. The presence of obesity and hyperleptinemia in the LRB mice prompted to test whether these mice were leptin resistant. They performed several experiments. First, they tested for leptin sensitivity in the LRB mice that remained lean throughout their lifespan. They restricted the amount of food the mice could eat so they could not become obese, and they normalized the plasma leptin levels. The leptin-evoked activation of hypothalamic STAT3 was significantly reduced in the calorie-restricted LRB mice as assessed by Western blotting. Second, they tested sensitivity in young LRB mice before development of obesity and hyperleptinemia. Leptin decreased food intake and body weight in young LRB mice, but only a small amount compared to the control. Data demonstrate that leptin resistance in mice lacking the BBS gene in LRB cells is not secondary to obesity. In conclusion, mice lacking the BBS gene in the LRB cells showed that obesity develops as a consequence of both increased food intake and lower energy expenditure. Data also revealed that these mice have reduced leptin sensitivity, and LRB signal transduction is a manner independent of obesity. So, a treatment for Bardet-Biedl, there's actually no cure for this syndrome, but there are some treatments for it. There's standard care from an ophthalmologist is required for management of refractive errors and assistance with low vision aids. As vision decreases, eventually they do lose all vision. Abnormalities of fingers and toes can be corrected surgically. Surgery can be performed to correct certain genital abnormalities and heart defects. Measures to manage obesity include age-appropriate diets, exercise programs, and behavioral modifications. In addition to affected individuals should be evaluated by a kidney specialist to determine effective intervention for kidney disease. Hormone replacement therapy may be necessary for males with hypogonadism. Services that may be beneficial may include special education for children with cognitive impairments, special services for children with vision problems, and other medical, social, or vocational services. Genetic counseling may be beneficial for individuals and their families. That's my presentation.